This is Group 4's Lab Report 5 on Biochemical Test 2 by Vianka Sanchez and Perez. The IMVIC tests, which include the indole test, the methyl red, and the Voges-Pressgauer, as well as the citrate test, are necessary for differentiating bacteria species, especially those within the Enterobacteriaceae family, by analyzing their metabolic characteristics. In order to diagnose illnesses and comprehend microbial behavior, microbiologists use this test to identify organisms by analyzing how they ferment glucose, use carbon sources, and break down amino acids. Materials. For the indole test, we use E. coli, E. clocae, unknown 204, 3-tryptophan broth tubes, an inoculation loop, and Covax reagent. For the citrate test, we used E. coli, E. clocae, unknown 204, 3 citrate agar test tubes, and an inoculation needle. For the methyl red, we used E. coli, E. clocae, unknown 204, 3 MRVR broth tubes, an inoculation loop, and methyl red. And for the Voges-Pressgauer, we used E. coli, E. clocae, unknown 204, 3 MR VP broth tubes, an inoculation loop, and VP1 and 2 reagents.
So for the methods, first of all, for the endo test, a sterilized test tube containing 4 milliliters of tryptophan broth was prepared. The broth was inoculated aseptically with growth from an 18 to 14-hour culture. So once we did all the procedure, we inoculated it. We left it in the incubator at 37 degrees Celsius for 20 to 28 hours. And at the next class time, when we recovered those results with some growth in them, we added zero point 0.5 milliliters of or 3 to 5 drops of Kovac reagent to the broad culture. And the presence of a red ring at the surface would indicate a positive result for the endo production. And then in the methyl red test, the methyl red broth is inoculated with the designated organism and incubated at 37 degrees Celsius for 20 to 24 hours. At the next class session, we drop 1 or 2 drops of methyl red reagent, which, if the organism has produced a stable acidic end products from glucose fermentation, the broth will turn red, indicating a positive result. For the Bose pros hour or BP test, BP broth was inoculated with the specific organism and incubated at 37 degrees Celsius for 24 to 28 hours. And in the next class, we added 15 drops of BP1 reagent and 5 drops of BP2 reagent. And the presence of a red color indicates a positive result, meaning the organism has produced acetylene as a neutral end product of glucose fermentation. Lastly, in the citrate test, Simmons citrate agar is inoculated lightly on the slant by touching the tip of the needle to an 18 to 24 hours old colony. The inoculated tube is incubated at 35 degrees Celsius to 37 degrees Celsius for 18 to 24 hours. A positive result is indicated by the development of a blue color on the agar gland, which means the organism is capable of utilizing citrate as its sole carbon source.
These are our results for the indole test. So E. coli was our only positive indole test because of the pink reddish color it has at the top. And the E. clocae and an unknown 204 had no change in color, which indicates a negative indole test. For our Voges-Pressgauer results, our E. coli and E. clocae came out positive, while our unknown 204 came out negative.
For the results of the citrate test, you would have on the left side of the slide pictures of the front and the back of this land, and it's as the order as it goes. So first E. coli, then unknown 204, and then Enterobacteria cloacae. First, let's start with E. coli. This test tube shows some growth, but the color didn't change, remaining green, which indicates that E. coli could grow in the medium, but it does not use the citrate as its sole carbon source, leading to a negative citrate test result. Second, we have unknown 204. These tubes show broad growth and a color change to blue, indicating a positive result, which means that the organism is capable of utilizing citrate, suggest, suggesting that unknown 204 is a citrate positive organism differentiating from a species like E. coli. Lastly, Enterobacteria cloacae, we could see like little minimum growth, but we don't know if that's growth or just the marks of the loop that was used to inoculate. So we also see that the color remained green, which makes kind of the result kind of like inconclusive, because supposedly Enterobacteria cloacae, it utilizes citrate, as it generally known to be a citrate positive, which usually comes back with a positive result. So for this procedure, we will have to check more for it, but it could be either wrong safety techniques to inoculate it, or it could be that the bacteria had something wrong in it. For the methyl red results, we also have the same dynamic of the pictures on the left, and they're the same order as we talked about them. First is E. coli, then Enterobacteria cloacae, and then lastly unknown 204. So first, the methyl red test is a red color at the top indicates positive results, meaning that the organism performed mixed acid fermentation and processes stable acidity and products from glucose fermentation. By knowing that, when we analyze E. coli, we see that it has some growth, and it also showed a red color at the top after the methyl red reagent was added, confirming that it's a possible result, which is expected because E. coli usually performs mixed acid fermentations. Then we have Enterobacteria cloacae. Similarly, this organism also showed a red color at the top, indicating a possibly methyl red result. However, Enterobacteria cloacae is more commonly associated with negative results for this test, so it may warrant further verification, as it generally favors butanol fermentation detected in BP tests. This would also confirm that something is wrong with it or Enterobacteria cloacae, because also on the citrate test we were supposed to see a positive result, but in this case we saw a negative, and over here we were supposed to see a negative result and we saw a positive. Lastly, or unknown 204, this organism displayed cloudiness growth in a red color at the top, indicating a positive methyl red result similarly to E. coli, suggesting that 204 may also carry out a mixed acid fermentation.
Conclusion. So the indole test is used to determine the ability of a bacteria to produce indole from the amino acid tryptophan. And after adding the Covax reagent, if the indole is present, it will turn into a red-pink color, as shown with our E. coli results, which indicates a positive test result. And a negative test result will have no color change, like the E. cloacae and unknown 204 showed. For the Vogus press scour, this test is used to test if the bacteria can produce acetonin, and it helps identify the bacteria, especially those in the Enterobacteria C family. So a red pinkish color indicates a positive test, and if it has no color change, then it's negative.
In this section of conclusions, we will talk about the citrate test and the methyl red test. First, let's start with the citrate test. In this test, E. coli demonstrated growth but no color change, confirming that it's a negative result as E. coli cannot use citrate as its sole carbon source. Then, unknown 204, on the other hand, showed both growth and a blue color changing, indicating a positive result and its ability to utilize citrate. For Enterobacteria cloacae, the result was inconclusive because of the potential procedure issues that might interfere with a typical positive result expected for this species, but unfortunately, we got a negative result. Moving on to the methyl red test, all three organisms, E. coli, Enterobacteria cloacae, and unknown 204, show a red coloration at the top of the tubes, indicating a positive result. This suggests that each organism performed mixed acid fermentation. However, the positive result for Enterobacteria cloacae is unexpected, as this especially a species typically shows a negative result due to its preference for butanol fermentation, which would be seen in the BP test. This anomaly may require further investigation to confirm the accuracy of the result. for Enterobacteria cloacae. But as I previously mentioned, Enterobacteria cloacae also was supposed to show a positive result on the citrate test, and it didn't. And apparently, we were not the only students that were having trouble with this bacteria. So probably the bacteria had some issue in it that interfere with the procedure being fully exact. But the overall, the citrate and methyl red test suggests that unknown 204 shares metabolic traits with E. coli, such as mixed acid fermentation, but differs in its ability to utilize citrate.